You're listening to the Future Tech Health Podcast with Richard Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that had, has, or will have them in the future. Medicine and biological systems are the final frontier. Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoy the podcast, please listen, subscribe, like, and share it with friends. Thank you.
Hello, this is Richard Jacobs with the Future Tech and Finding Genius podcast series. I have Herve Tiriac. He's an associate project scientist at UC San Diego Health. And we're going to talk about developing precision medicine platforms and specifically 3D organoid modeling, patient-derived organoids, which I've spoken about a few times. So, Herve, thanks for coming.
Thank you so much for having me. I really appreciate it.
Yeah, I don't know if I think I've asked this before. So, patient-derived organoids. Can you just describe that process? And if it's not patient-derived, where do these cells come from to make an organoid?
Yeah, so the term organoid is actually quite an old term, but recently has been associated with the methodologies that have been developed by Dr. Hans Cleaver and Dr. Toshi Sato, who have worked together back in the early 2010s. And they published some really nice series of papers demonstrating the procedures, the protocols, and the methods for growing three-dimensional organ-like structures, initially from the intestine and then later on from other organs. And in collaborating with Dr. Cleaver's, my previous mentor, Dr. Tuvison, they collaborated and helped establish the initial methods for the pancreatic organoids. So everything that I have done in the Tuvison lab and now independently at UC San Diego has been starting from adult tissues, predominantly human in my case, but we can also do it from the mouse models, for example. You can also make organoids from iPS cells, so starting with the stem cell and differentiating into a specific tissue lineage and maintaining it in an organoid-like fashion. But that's not really my specialty. It's sort of a different track. That's the engineering track, I would guess.
So you were focused on deriving pancreas-like organoids?
Yes, my specialty has been pancreatic cancer, specifically pancreatic ductal adenocarcinoma, for both personal and scientific reasons. It's a very deadly disease. It's also still understudied and has a lot of potential for leading to improvement in the clinic. So we would like to make an impact to the research.
So in terms of the pancreas, I mean, I know an organoid approximates the function of the organ, but how many different functions and different cell types and different parts of the pancreas can be modeled successfully in an organoid? What do you pick?
Yeah, it's a very good question. So indeed, the organoid system initially was described as containing all the relevant cell types from the organ from which it's derived. And that was the intestinal organoid. The pancreatic organoids, however, they're specifically modeling the ductal cells, the epithelial ductal cells from the pancreas, which we think are the precursor for the cells that lead to cancer, to ductal adenocarcinoma. So we're not modeling using the ductal organoids. We're not modeling the acinar exocrine compartment, the one that's secreting all the enzyme that digest your food. And we're also not modeling the neuroendocrine cells of the pancreas, the islet cells that generate the insulin and other hormones. So this is very specifically looking at ductal systems, and we are studying both normal as well as pre-neoplastic and neoplastic, meaning cancer, in that system.
Well, I mean, do you think that you'll be missing out on potential effects? I mean, I would think the pancreas is very coordinated, like any organ system, so that all the different parts talk a lot and have a lot of interaction.
Yeah, absolutely. The ideal scenario is to have all the compartments present when you're modeling something. However, the initial first step was to develop the methods for specifically modeling the normal and the transformed ductal system. Now that we have that, we're starting to put in back additional compartments, specifically the mesenchymal compartments, or the fibroblasts that surround the cancer cells, as well as immune cells that are also a major part of the microenvironment of the tumor. Now, to go a little further from there, it would be ideal to also have the acinar and the endocrine cells present in the same culture, but that becomes a very complex culture, and you have to remember that to support the growth and propagation of these cells in vitro, you have to provide them with the media and the cues for their survival and proliferation. And those are not exactly the same for epithelial cell, mesenchymal cell, endocrine cells. So we can probably find a happy medium to keep everything in culture, but I don't think that has been described yet, at least not for propagation of the model.
Do you see any remodeling going on once cells are in culture, especially if you have multiple types or a single type? Are they trying to establish structure?
Oh, yeah, absolutely. There's been some really nice work published by my former mentor, Dr. Tuvison, with some of my colleagues, that has shown very nicely that if you put in culture the ductal organoids with the cancer-associated fibroblast, they will form structures, and they will remodel both the cancer cells and the mesenchymal programs, so that they become basically established as a synergistic culture. So absolutely, there is a lot going on once you put different cell types in co-culture, and if they synergize, then they can remodel into different compartments.
So if you compare, a very primitive organoid with just one cell type versus the ones that you're trying to do now, the more complex ones, are you seeing very different behavior, or is the behavior staying pretty consistent?
So the behavior in terms of the genomics stays consistent. We don't see genetic drift, at least not over 10 passages or so. What does differ when you change the culture conditions is typically the transcriptome. And that sort of makes sense because you have now additional signaling from different cell types, and that's going to remodel the cell's behavior.
How do you study pancreatic cancer, for instance? So are you culturing just tumor cells, and that becomes your organoid? It's like a tumor-based organoid, or do you have an organoid system whereby one, just a part of that organoid that you have in culture is comprised of tumor cells?
Oh, I understand. So the process typically starts by acquiring the tumor. So this is patient, tumor. And then the tissue is dissociated into small fragments, not necessarily single cells. In general, actually, single cells sometimes is detrimental for the generation of organoids. And from these fragments, we see organoids form. Now, at this point, what we would like to do in the laboratory is to isolate just the tumor organoids and sort of preserve those as a tumor-only culture. And if there's normal organoids also present, we'd like to also separate those and have a normal culture that's free of tumor. So in general, our cultures are either tumor or normal. They're typically not mixed. And we know that because when we sequence the organoid, if it is tumor, we only see the mutations that are present in the tumor, and there doesn't seem to be any contribution from normal cells. And on the other side, from a normal culture, we don't see any neoplastic mutations. We don't see any transformation of the cells. So the cultures are independent, and as far as we can tell, they are 100% pure tumor or normal. And that's sort of by design, and it's also because the media conditions that we're currently using are promoting only the growth of this epithelial ductal system. So the other cell types that are present in the tumor, being fibroblast and immune cells, they're not able to proliferate and maintain themselves in culture. So within one or two passages, all these other contaminants are basically gone. and you're just left with the ductal, either normal or tumor cells. We typically make sure that we're starting only from tumor tissue when we're trying to obtain a tumor organoid by starting with tissue that is specifically taken from, close to the center of the tumor, not too close to the edge, where you potentially have some mixing of normal and tumor cells. And same thing with the normal cells. If we're trying to make normal organoids, we'll start from a piece of tissue that's removed from the tumor where there's a less likelihood of having contaminating tumor cells present.
Well, what's wrong with having a mixture? I mean, in the body, you can't avoid having a mixture, and you can't avoid having the interaction with other, for instance, pancreas cells that are not tumor cells. So is it just,
absolutely.
it would make it difficult to get the right data and do the right testing if you had a mixture? Is that why?
Yeah, so it's partially a question of how do you preserve a specific ratio if you're going to maintain the two of them in culture for a long term. So to back up for a second, initially we try to establish the models independently, characterize them, and basically have tumor versus normal separate. But we are able to put them back together at specific ratios, so we can do 50-50 mix in culture, and we can also label the cells so we know what we're looking at. So we can transform the tumor organoids to be red fluorescent and the normal organoids to be green fluorescent, for example, and put them in culture and be able to actually tell which one is a normal cell versus a tumor cell. But if you maintain them from the get-go as a sort of a mix, my guess is that this ratio wouldn't stay constant. It would keep evolving over time. And one interesting fact of the organoid methodology is that typically normal cells are able to divide faster and are more successful in their division because the tumor cells are typically aneuploid. So they don't have equal genomes to divide at every division, and not every division is successful for the tumor cells. So if you have both normal and tumor cells, at least for pancreatic ductal adenocarcinoma, in culture at the same time, and you keep passaging, over time, the expectation is that the normal cells will outgrow the tumor cells.
Oh, well, it's funny. I thought the tumor cells would outgrow the normal cells, I guess, because supposedly they divide extremely fast and they're aggressive, right?
Yeah, but you have to remember that in the organoid system, the normal cells don't behave like exactly the same as the normal cells in your pancreas, for example. They're not quiescent. They're not. They're replicative in this instance. So it's more similar to an inflammation response where the normal cells have to divide to do some wound healing. And in that case, normal cells can divide really fast because, as you know, we heal pretty quickly, actually.
Okay, I see. So what are you trying to figure out? I should ask you this first. What are you trying to study and figure out by making these these tumor organoids or normal cell organoids? Yeah,
so the major goal of my research is really to move toward precision medicine using these models with the expectation that making these models from patient would help us better understand the biology of the tumors. And we know that every patient is different. And we've observed that every model that we've generated is quite different in its response to, for example, standard of care chemotherapy treatment. So we think that having a model for the patient would help us identify the right treatment for the patient. But beyond precision medicine, having these models is extremely useful for research because we're now talking about looking at cohorts of models of, close to 100 models now instead of just of a few cell lines to represent the entirety of pancreatic ductal adenocarcinoma. So we have a much better representation of, the different types of cancers that are seen in patients, both early cancers and late cancers, because using organoid methodology, we're able to make models from fine needle biopsies, for example. So we don't need a lot of starting sample to make these models, which enables us to study really late cancer where all you can get is a little tiny needle biopsy from a metastatic site, for example.
So what are you seeing? What's the difference in the models? You said there's many different kinds of, of cancer types. What kind of variation do you see?
Yeah, so the first variation is at the genomic level. So in pancreatic cancer, we expect that the majority of the models will have a KRAS mutation, and that is true. But after that, you're seeing a lot of p53, and then it becomes very split. So there's a lot of mutations that are model specific. And we're not sure if those mutations are impacting the response to therapy, for example. So that's one ongoing area of research. I think the major difference that we've seen that's been extremely interesting to study has been the response to chemotherapeutic agents, for example, where we've tested a cohort of close to 70 different patient-derived models using the standard of care chemotherapeutic agents that are used in the clinic for pancreatic ductal adenocarcinoma patients. And we're observing distinct responses from very resistant to very sensitive for each agent. And we're seeing patterns where some models are very sensitive and some models are very resistant to all therapy. And then there's a lot of models sort of in between that that are sensitive to maybe one agent but resistant to the rest of them. Yes?
One question here. I mean, from what I've heard, cancer is very heterogeneous. So
that's right.
If you see something that has a P53 mutation, that could probably only mean it's the predominant one. It doesn't mean it's the only one. And it probably has KRAS too, just maybe in a lower amount, and vice versa for the KRAS predominant one. So I would think that would definitely confound the results unless you looked at all the possible mutations and, sorted them kind of like in a Pareto and saw which ones are predominant. Because from conversations I've had, it seems like they're all there. They're just there in varying amounts.
Right. So you're talking about associating the mutation frequency, the allele frequency of a mutation to a specific therapeutic response, correct?
Yeah, I think that, because I guess that's more of a true picture of what's really going on, right?
Right. So that's a fantastic question. And in fact, we haven't been able to associate general sensitivity or resistance to treatment with those mutations. And the issue, like you mentioned, is the majority of the patients are going to have a KRAS and a P53 mutation. So it's not, there's not enough either samples or there's not enough diversity in the mutation profile at that top level to really be able to associate that to a therapeutic response. What would be interesting is to find these passenger mutations, or I guess maybe they're not passengers necessarily, but they might not be the main drivers of cancer, like KRAS and P53 and our models, that may give a sensitivity to a specific agent. So that's more what I'm talking about, not necessarily association with KRAS and P53, because again, we've looked and we haven't seen those specific correlation to sensitivity or resistance with just those mutations.
Well, also too, if you expose a tumor organoid to various chemotherapy protocols, the ones that don't completely kill it, what happens then? Can you observe the development of resistance and the new mutations that are becoming predominant, or there's a selection process by which now, okay, this mutation was rare in this previous sample, but now it's predominating after this chemo treatment.
Mm-hmm. Yeah, so that work is ongoing. All the resistance and the sensitivity that we've described so far has been pre-existing, meaning it came with the model. And we're now trying in current studies to see if we can select resistant clones from a pre-existing organoid and profile those clones and see exactly what happened, like you're saying, which mutations have been enriched for and how did this resistant clone become resistant. So that's a great question, but I don't have an answer for you right now on that question. However, these models are definitely usable for that type of study.
Well, I guess to make it even worse, I don't know how this plays in, but I guess I keep referring to it because it was really interesting. I spoke to Florencia McAllister, and she's studying the microbial constituent of pancreatic tumors, meaning that they have their own microbiome. So in these models, I would guess no microbes are included, certainly not deliberately, but if you were to, would that completely change the model and how it works? Yeah,
that's a great question. And that area of microbiology and how it actually affects the tumor's behavior is of high interest in the field right now. And yeah, absolutely, you're right. We're not including those microbes in the current cultures. It would be quite difficult to maintain them over multiple passages without having, gross over-contamination and having to get rid of the culture. So that's sort of an ongoing potential area of research, not necessarily for myself, but in the field. The interesting observation that we have is some models are unfortunately contaminated pretty much within one passage after establishment. typically with yeast, but sometimes with bacteria. And the current question is that something that was pre-existing in the patient tumor, or is that a mistake on our part and some sort of cross-contamination with something? And that's unclear at this point. It would be really interesting to have sort of a parallel generation of organoid models and try to generate whatever microbes and fungi cultures are pre-existing in the tumor. However, that's not going to be trivial.
That's true. So when you say your model gets contaminated by, I guess, bacteria, fungi, yeast, etc., what happens to the organoid? Does it just die typically, or does it still maintain and live on, just look different? Like, what happens?
Yeah, so that's a rare occurrence. Luckily for us in generating the models, I would say maybe 10% or less frequency, and it typically happens within a few days after processing the tissue from the patient, and you get gross overgrowth of typically fungi or yeast, and you lose the model pretty quickly. So they're not able to sort of maintain a sort of a healthy ratio for both cell types.
Gotcha.
So for us, it's a complete loss. Yeah.
Okay. Interesting. Yeah, there's so many experiments to do. It's impossible. There's only so much you can do.
There's a lot of things that are possible. So the first thing that we focused on is just making the models, characterizing them, trying to demonstrate for ourselves and for the community that they are representative of the tissue from which they're derived. And I think now that we have done, hopefully, a good job at that first step, we're going to move on towards the more interesting experiments, which involve the co-cultures like we talked about earlier.
Yeah, so what specifically are you hoping to figure out with your new experimentation?
Well, one goal is, like you mentioned earlier, is to understand resistance to treatment. We know that most of the patients with this disease are, even if they're responsive to a treatment, they're going to relapse eventually. And we don't necessarily understand all the mechanisms by which they become resistant and how to prevent that resistance from being an issue clinically. So I would say that's my sort of primary research focus. And the secondary research focus is more engineering and protocol driven in trying to sort of better the protocols to be able to generate these organoids faster, at higher efficiency, and also to be able to generate the cell types, the other cell types that come from models for the other cell types that come from the tumor, such as the cancer-associated fibroblasts and macrophages and other cells that we can obtain from the tumor, so that we can then reconstitute a more complex co-culture system at a later time. So it's both a science question and sort of a engineering question at this point.
And with the current models you've developed, any interesting or inexplicable behavior or results you're seeing so far?
Well, there's a lot of variation from model to model, even starting with the morphology. A lot of these are sort of round, cystic, three-dimensional objects, but sometimes they're filled, sometimes they have sort of interesting structures that we can't really explain at this time. So there's probably too much variation for us to really be able to be too surprised by one single phenotype. But I guess one of the most surprising findings that I've had, at least for myself, was when we did the transcriptome analysis of our cohort. And again, this was 40 or plus models. We noticed that there was very good representation of both the classical and the basal subtype of pancreatic cancer that have been previously described by other groups in the organoid models. And I was pleasantly surprised by that result because all of these organoids are grown in the exact same condition. So same media, same matrix, same everything. And the one possibility was that the milieu in which we're growing the organoids would reprogram the cells in such a way that you would lose that subtype specificity from the patient. And it appears, at least so far, that we're maintaining that subtype specification in the organoid, which is good for research and for really having representative models to study.
What does it look like for early stage cancers versus very late stage? What kind of differences do you see?
So the late stage cancers that we have mostly come from these liver mets or sometimes lung mets, from which we've obtained small biopsies and generating the models. And those are clearly more aggressive. They appear to be generally a little bit more filled and solid. They are typically much more resistant to the chemotherapeutic treatments. However, they're not completely resistant. We often find some drugs that are very efficacious in killing the models, even if they're advanced. And they seem to grow at about the same proliferation rate as the early cancer models. On the other side of the spectrum, these early models, and I'm putting sort of air quotes on that, they tend to be a little bit more cystic and generally more responsive to treatment than the late derived models. In terms of mutation profiles, we see a lot more mutation burden in the advanced models than the early models, and that makes sense. I think one of the issues in pancreas cancer is that with a late diagnosis, most patients are diagnosed with significant tumor sizes already. So if you were to look at our cohort, even though we have 60 plus models, I think out of the 60 tumor models that we have, only 2 are derived from stage 1A and 1B tumors, because those are extremely rare diagnosis events and resection events. So we do have these early models that have been resected, that have been removed by a surgeon, but I would say they're probably not the earliest type of cancer that we could really be studying in the laboratory. So it's still really important that we accumulate more models from these really early patients. So, true stage 1 patients to really compare to the late metastatic models.
A question, in the metastases, are they composed just of pancreas type cells? I just wonder if when a cancer metastasizes, does it ever form a chimera, where it somehow corrupts the cells that it's taken root in? You said pancreatic cancer, where does it metastasize to typically?
The typical metastatic sites are the liver, and then that's followed by the lung. And initially, I would say in the really early stage cultures, you probably have some normal liver cells and normal lung cells that would persist in culture. And we've had actually really interesting observations. So for example, one of our early lung metastasis-derived organoids, we've noticed a few cystic organoids in culture, in really early cultures, that were actually ciliated and spinning around. And those were probably derived from the normal lung epithelium and not actually cancer. However, upon passaging, we basically lost all of those ciliated organoids, and we're just tumor keras containing organoids. So by the time we characterize these models, for example, in that model, I believe p53 was at 100%, p53 mutation was at 100% allele frequency, which indicated that you really didn't have any normal cells left.
But I guess, I don't know if, I'll just very bluntly, if a pancreatic cancer metastasizes to the liver, is it able, through cell-to-cell communication, exosomes, whatever, is it able to turn some of the liver cells cancerous, I guess, to put it bluntly? So does it create a metastasis that's composed of 2 different organ type cells, or is it still pancreatic cells that are just set up shop in the liver?
Yeah, so you're asking me a question for which I'm not equipped to give you a proper answer. As far as I know, that's not been demonstrated, but I am not an expert on that, on this field. I don't think you can, transform another cell through some sort of, communication.
Well, the reason why I ask is because from what I've read, the exosomes released by, like a primary tumor are able to at least set up a niche, set up a preferential place by which a metastasis can occur. So they're at least somewhat changing the recipient cells, the liver cells, for instance. But I don't know if they're going as far to turn them into cancer cells and change their genes and everything and rearrange their DNA. It's a big leap, but have you ever heard of such a thing, or no one knows?
So I don't think, again, on the transforming, meaning like altering the genome of a adjacent cell, I'm not sure that research evidence exists right now, but there is actually good evidence, as far as I know, for reprogramming the microenvironment of a tumor. So if a tumor cell from the pancreas seeds in the liver, it's able to over time, reprogram its environment to support the tumor growth. And we know that that doesn't happen instantaneously. It takes some time for that tumor cell to reprogram its environment. And there's great work coming from multiple labs that are sort of dissecting how the tumor cells are able to reprogram their environment, which I'd be happy to share with you later. I think if there is any example of sort of cross transformation across cell types, it would probably be in viral driven cancer models. But again, that's not really my specialty, so I can't really speak on that myself.
No, it's okay. It's just something I thought of, and as I'm happy to hear that it doesn't seem to be that way. Thank God.
Yeah, it would be. It would be dramatic if tumor cells were able to do that, for sure.
Okay, well, very good. Well, it's a difficult job, I'm sure, for you to develop these models because, like I said, there's maybe microbial stuff going on, exosomes that are coming out or going in, different cells signaling to each other. I mean, it's just endless. So I guess in one way, it's amazing the models work at all. But
right,
thank goodness that they seem to. So that's great.
Right. So one of the things that we were aiming to do with our initial studies in the Tuvison lab was to demonstrate that these models actually were able to recapitulate a phenotype that was observed in the patient. So, and that would be a cancer intrinsic phenotype in this case, because we're only looking at the cancer cells and the organoid again. And the way we were able to sort of demonstrate that for ourselves was in sort of a two-pronged fashion. The first one is we had organoid models from patients from which we really had good knowledge about their clinical outcome. We knew which treatments they responded to and which treatments they did not respond to. And the organoid models tended to demonstrate similar responses when tested ex vivo with the same agents, meaning if the patient was sensitive to a specific treatment, the organoid tended to be more specific to that type of treatment, and vice versa. And then the other way we're able to demonstrate some sort of predictability from the organoid to the patient was to extract transcriptomic biomarkers of sensitivity from the organoid cultures. So using all of our testing with chemotherapeutic agents and all of the RNA sequencing we had done on the models, we did a correlation between these two data sets, and we pulled out gene signatures that were correlated with sensitivity to chemotherapy and gene signatures that were correlated to resistance to chemotherapy. And then we applied those same gene signatures to patient cohorts from which we did not have models, but for which we knew the outcome. And were able to statistically show that there was increased responses to treatments and increased survival for the patients that actually had the sensitivity transcriptomic biomarkers. So it's sort of another method that demonstrated to us that the organoids predicted, at least in part, the response to therapy. Now, again, I don't think that organoid by itself, cancer by itself is going to be able to tell us the whole picture. We're going to need to put in these other cell types we talked about to really get a more complete picture of what the tumor will respond to and how it will respond to the treatment. But at least this first step was successful, which is important for the next step.
Okay, well, excellent. Well, very good. Herve, what's the best way for people to find out more, read some of your papers and interact with the lab or you?
Podcasts like this one are a really good way to interact and learn more about this science. But beyond that, there is quite a lot of great resources online for learning about this cancer and about the research that we're doing. There's two great foundations that work in pancreas cancer, the Lustgarten Foundation and the Pancreatic Action Network, or PanCAN. They both have fantastic resources for patients and also for people that want to learn more about this. And if you want to do a deep dive on the topic, I suggest you head to the PubMed website and just start Googling some names or some questions and start pulling out some reviews or perspectives on the research. And those are usually a good introduction to the research that's ongoing. And from there, you can jump in the primary research articles that contain all the data that we've published so far.
Okay, excellent. Well, Herve, thank you for coming. I appreciate it.
Thank you so much for having me.
You're listening to the Future Tech Health Podcast with Richard Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that had, has, or will have them in the future. Medicine and biological systems are the final frontier. Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoyed the podcast, please listen, subscribe, like, and share it with friends. Thank you.